everybody. I hope you are doing fabulously this Tuesday morning. We are so excited to work with you all and to explore the wonderful world of algae with you. Before I hand things off to our presenters, I just want to get everyone situated using the Zoom chat function, because that is how you can ask us your questions. We do have one of our specialists watching the chat, so if you have any questions, please submit those. And to get everyone comfortable using it, go ahead and navigate to the little chat bubble at the bottom of your Zoom window. If you click that, you'll be able to type in a chat and just make sure that it is set to send to everyone. I'm mostly asking that so, people know what questions are being asked. It doesn't look like our presenters are talking to themselves. It just makes things a lot smoother. So go ahead and let me know what you are up to this summer. So in that chat bubble, are you hiking this summer? Are you teaching summer classes? What is the big focus of your summer month? I know not everyone's done with school, but some folks are wrapping up. Ooh, looks like we've got some folks traveling. Good for you. I am jealous. Got folks working. Yep, staying cool. I love that. Cleaning. Hanging out, I love that. Taking classes, good for you. That is awesome. Very nice. Well, while we still have some folks coming in, I will just hop to some housekeeping items, just to make the most of our time together today. So one of the most important things for you to know is that you will get a recording of this webinar. It will be live both on our website and our YouTube channel. It usually takes about 3 to 5 business days for us to edit that down and get it live, but as soon as it's ready, we'll email it to you. We will also have a folder with additional resources and a certificate of attendance in that email. I'm also going to drop it in the chat box here in just a second when I'm done talking, so keep an eye out for that. And as I mentioned before, we would love to answer your questions during today's session. So please use that chat bubble at the bottom of your Zoom window. Set that setting to everyone so we can all see your question and let us know what questions you have. We'd love to answer it today. And if we happen to not get to your question, please feel free to reach out. We are more than happy to help you out. I've got some of our email addresses on the bottom of that screen there. With all that being said, I'll go ahead and pass the presentation off to our folks, and thank you again for joining us, y'all.
Hi there. So what I was going to do, my name is John Melville. I'm going to take you through our today's agenda and then basically introduce everybody, but get us all on the same page. So the first thing we're going to do is you're going to meet all of us. You're going to get to meet me, Sarah, and Matt. I'm going to do a brief introduction to our sensors and software. We'll talk about some amazing resources from Algae Research Supply. Then I'll talk to you a little bit about our primary productivity activity. It's a very straightforward primary productivity activity that uses our DO probe. But probably even more exciting is we're going to talk about how to measure aquatic photosynthesis and cellular respiration in real time using real algae from Algae Research Supply and our DO probe. And like I said, questions are welcome at any time. So just quickly about me. My name is John Melville. I'm the College Biology Relationship Manager. I have a bachelor's degree in biology and psychology from Sonoma State University, where interestingly, my focus in biology was marine science. I then got a PhD in zoology at Oregon State University, did some postdoctoral research at University of Oregon. But most important for this discussion is I was a college professor at a little place called Wartburg College and also at Lane Community College. And almost all of my courses were majors and non-majors courses in biology, neuroscience, and also marine science. But I really like to focus on active learning with an emphasis on data collection and definitely inquiry. I'll hand it off to Sarah here. Oh, you're muted, Sarah.
Oh, that's odd. I clicked on it and it didn't unmute. My name is Sara Tallarovic. I also have a PhD from Oregon State. in zoology, and I was a biology professor for about 15 years in South Texas. And I taught everything from intro bio to human anatomy physiology. And then my first love, which is animal behavior, courses in neuroendocrine and behavior and neuroscience. And I also have a background in active learning and cooperative learning in laboratory settings.
All right, Matt.
Cool. Can you guys hear me okay? Awesome. Cool. Well, do I need to switch anything over so that I'm on the camera part or whatever? Or just my voice is ethereal? I will let that be what it is. Well, my name is Matt Huber. I am a biological oceanographer. I started, since we're giving a little background on ourselves, I started my career out doing pharmaceutical research. I worked for Allergan, helped get Botox approved. I changed directions entirely and got into natural science because I wanted to save the world from carbon dioxide. And I did just that. I went and did my graduate work on the relationship between aquatic photosynthesis and levels of carbon dioxide. From there, I went and did mass cultures of algae. I worked for giant spirulina farms out in the Imperial Valley of California, as well as I got involved with the algae biofuels programs. And I built large algae biofuels projects in California, built a few of them out here in Texas, New Mexico, and Hawaii. And after that whole algae biofuels bubble popped, I really wanted to stick with my core belief that we can do something. And my wife gave me the idea to start putting our kits onto, or put algae teaching materials on Amazon. And so after so many years, I figured out that it might not be me that comes up with the giant ideas to save the world from CO2 with my giant algae biofuels project. More likely it's going to be the teachers, and more likely than that, the students who need the information that myself and the people I know have. And so we freely want you guys to have that knowledge so that you guys have the tools. I want you to wield algae as the tool to fix climate change. That's why I'm here.
Yay. And one of the great things that we found in working with Matt is that his Algae Research Supply company has amazing cultures of algae, all different types of algae, and even algae beads. And what's great is that you can use our sensors with his resources. So what I wanted to talk to you briefly about, or show you really quickly, is we have some of our core sensors that I'm showing you right here, the Go Direct CO2 gas sensor, which cannot be put in water, the Go Direct O2 gas sensor, which also cannot be put in water. But the Go Direct pH sensor and our Go Direct optical dissolved oxygen probe can be put in water so that you can monitor cellular respiration and photosynthesis. And they're really great to use. They all use these Go Direct sensors, do not use Logger Pro. They use graphical analysis. Graphical analysis can be installed on any device. It can be installed on a Chromebook, Mac, PC, Android phone, tablet, iOS device, etc. One of the great things about the Go Direct optical dissolved oxygen sensor is, like I said, it's useful for aquatic photosynthesis, aquatic cellular respiration, biochemical oxygen demand. That's what BOD stands for, and also primary productivity. It's also great to use in water quality and environmental science. And
John, can I interrupt you on your dissolved oxygen sensor there?
Yeah.
For those of you guys who have ever had to use a DO sensor that had like the little membrane, the little plastic thing, and you had to get the liquid inside it and get the bubbles out, and then it didn't work right, so you had to recalibrate it. Dude, this thing is, I've had to work with DO sensors for about 26 years now. And finally one, it's super easy to use. There's no crazy chemicals that you got to make down or buy to do stuff with. It literally just works out of the box. It makes it so much easier to do DO in water. And oxygen or pH probes, those are about the same. They work really well here. But the DO probe, holy moly, this is, I'm over the moon for it. We're going to go over some data here in a sec.
That's awesome. So
sorry, John.
No, that's great. That's great. I love it. So one of the other really cool things about our DO probe is it has multiple sensor channels. So the DO probes have multiple sensor channels. So it can measure percent saturation, dissolved oxygen, but it also is a temperature probe and has salinity correction in it already. And just to explain a little bit how it works, the reason why it's so great and amazing is because it's has fluorescent material in the cap that is can be referred to as a luminophore. There's a blue LED that excites it, so when you plug it in, it'll shine blue, red, blue, red, blue, red. So the blue light hits the cap, it then emits red light, so it fluoresces, and the amount of red light that it emits is dependent on the amount of oxygen that has diffused into the cap. And there's two ways that you can actually measure the amount of dissolved oxygen. You can either look at the intensity of that red light, or you can look at the time difference, and that's what we use. So if you're like a real like nerd geek like me and you want to know how all this stuff works, this is like a little diagram of like what's inside the probe itself. The cap is right here. The luminophore is on the outside. There's a little filter in there. Then there's this blue LED. The light shines down, hits it, it bounces off and is detected by a photodiode. And then we also have a red LED that we use for reference that's also flashing at the same time. So it's really cool. And what I was going to show you next, I mean, we could talk about the pH probe, but what I thought we might show you, well, let's talk about the pH probe too, and then I promise we'll come back to the DO probe. The pH probe is also great because you can measure pH, use it for water quality, environmental science, but also, as Matt's going to show you, you can also use it for aquatic photosynthesis and respiration. We also have a TRIS compatible flat pH probe, which I think might be a little bit better in most cases.
That's what we have here. Yeah.
Yeah, and that's just because it's got a dual junction. It's going to respond a little bit faster, but either one of those would work fine. Let me just show you a live demo of GA so you can see what I'm talking about. There's GA right here. Oh, let me go out here for a minute and show you. Here's visualizer, and then I'll pull up GA. Where'd you go, GA? There you go. There. So what I've got right here is I've shown you graphical analysis, which is, like I said, our free software. And then here is the DO probe. It's in a little case right here with some water. I've tapped on this little button right here. Let me move over there. So if you tap on this button, it's actually already connected. I'm going to turn it off here because I already connected it to my phone. But if you want to just connect it directly, there is a USB cable. So you can plug it directly in using the USB cable, or you can connect it wirelessly. To connect it wirelessly, all that you need to do is turn it on and turn it off again here really quick. There we go. So now it's flashing red. I'm going to select sensor data collection. It's going to broadcast that it's a DO probe with this number. You can see that on the back, there's that same number right there. You wouldn't need to do any of this if you just connected it over USB, but I just want to show you this. And then right now I'm collecting DO. And like I said, there's different sensor channels. So if you want to measure saturation, temperature, pressure, you can. But it's that easy. I'm measuring DO, and now what I'm going to do right here is I'm just going to, I'll show you this right here. I can actually turn on a meter if I wanted to, just a meter, and turn off the graph. And I'm going to take the DO probe right here, and I'm just going to stick it in some water. It's actually in some water. And there we go. I'm measuring DO. It's actually that easy, just like Matt said. There's no other things to turn on, put in, etc. It's just amazingly simple to use. All right, so let me go back to our slide deck.
John, we had a great question in the chat,
Yep.
and it was whether graphical analysis has to be downloaded or does it run online? I answered it in writing, but I thought you might want to elaborate a little bit more on the versatility of the different ways that you could run the app.
Yeah, there is a way to run it. Hold on here. You can run it on a Chrome web browser. So if you just type in graphical analysis.app in a web browser like I'm doing right now, that will actually launch the app. So you can, if you have anything that's running, that if you can run Chrome, you can basically just launch the Chrome web browser, type in graphical analysis.app, and you will be running what we call our progressive web app. The connections are slightly different, but very similar. If you plug into USB, there'll be a step where you have to say, yes, I want to plug into USB, but you don't have to download it. You can totally use the progressive web app and not have to download any software. So that was the live demo. I want to make sure that I talk a little bit about our primary productivity lab, and then we'll hand it off to Matt so he can talk about what I call the exciting stuff. But there is a standard primary productivity lab in two of our books, Biology with Vernier, that just looks at the amount of light that there is from a sample, like how much primary productivity there is if you're blocking a certain amount of light. But even more interesting is I think, is we have an inquiry book, and this is experiment 17 from Investigating Biology Through Inquiry, where you can actually investigate primary productivity. So students can look at things like, well, what happens with different levels of light? What happens if you're using different colors of light? Or even more interesting that Matt could probably even talk to about is what happens if you add iron and does that increase primary productivity or other things like other nutrients, which would be really interesting. So both of those can be found in our book. The lab itself is very simple, but before I get to that, I just want to remind everybody that when we're talking about primary productivity, remember that I just want to bring this up because this was always the #1 misconception that my students had when talking about plants or algae is they would forget that plants and algae can perform cellular respiration. Yes, they have mitochondria, but then in the sunlight, they have chloroplasts, right, which will create oxygen and actually consume CO2 at the same time. So when you're looking at primary productivity, one of the things we can do is see does the amount of photosynthesis, is that greater than the amount of respiration, to see whether there's net productivity or just gross productivity going on. All right, so before we get into that, I just want to ask everybody to answer this in chat. What percent of atmospheric of O2 comes from the ocean? Anybody want to guess? Everybody talks about forests, stuff like that. I'm from the Northwest and Northern California. But does anybody want to tell me, give me an answer there? Just remember, there's answer E. I'm going to thank my professor, Professor Kelson from Sonoma State University, that loved to do this. He would put 2 items on a test that were both correct and then make you guess E. So E is actually the correct answer. It's somewhere between 50 to 75%, depending, or it could even be more. So, yeah, over half of the atmospheric oxygen that we breathe actually comes from the ocean or marine photosynthesis, which is, by the way, going to be produced by mostly algae. All right, so the standard way of looking at primary productivity is using what we call the light and dark bottle method. And that is where you just have a bottle that you put in light and a bottle that you leave in the dark, and you measure the DO before you leave them for 24 hours in the light and the dark. And typically this can only be done at certain times of the year because you need algae to be present, right? So like now it's June, you can go out and go to a pond or somewhere like that and grab an algae sample, or grab some water in your from a pond, and it's going to have algae or other stuff in it. The problem can be sometimes is it'll also have other microorganisms that are not photosynthetic, and so your respiration rate will be really, really high, and that can be confusing to students. What's great about Algae Research Supply is you can just buy chlorella from them directly, and then you can use that as your source. And that means that you can do this lab at any time of year, even in the winter, no matter where you are. Just buy some chlorella culture from Algae Research Supply. And you don't need that much. You just need to add 5 mils of that algal culture in 1 liter of water. Now, In our lab, what you typically do is you determine the respiration rate. And that's just if you have a bottle in the dark and then you have the DO that you recorded initially 24 hours before. The difference in those two will give you the respiration rate. Gross productivity is just going to be the DO of the sample in the light minus the dark bottle. So that's gross. Net productivity, though, is going to be the DO of your sample that's been in the light, for 24 hours, minus your initial DO, the DO before. Now, let me show you why that can get a little bit confusing in some cases. This is, I just did this experiment yesterday and measured it. I took a sample of water from Beaverton Creek. You can see that the initial DO at the end of the day was 9.8 milligrams of oxygen per liter. There's probably a little algae bloom going on there. And then I left them one in the light and put another one in the dark. And you can see that it dropped almost like a full milligram there. It went from 9.8 to 8.96 in the dark. And even the light went down, right? So the sample that was in the light, even that went down. So students tend to get confused because they would typically ask me, hey, Dr. Melville, that doesn't make sense. Shouldn't there be more oxygen for the bottle that was in the light? And then you have to get in this whole discussion of, well, there's other stuff in that bottle. There's microorganisms, there's zooplankton, there's probably bacteria. If you use the algae cultures, you typically don't get into that discussion. And it makes it a lot simpler for your students. And that's why I also think the resources from Algae Research Supply are amazing. Because in this case, if you look over at the algae culture sample that I used, the initial reading was 8.46 milligrams of O2 per liter. And you can see in the dark, it did go up a little bit, but that's because there was probably some residual photosynthesis happening while it was in the dark. But it should be really simple for the students to see that, hey, this number, the number in the light, is way bigger than my initial. And even more important, the number where it was in the light is still much greater than it was in the dark. So we can definitely see that there was not just gross productivity that happened, but some net primary productivity. So if what you want to do is drive home how important algae are to producing oxygen and then also fixing carbon, it's going to be a lot simpler for your students if you use algae cultures than a water sample from a creek or pond. Now, just one last little thing here, and this is in that lab. If you do it, there's a little link there that tells you how to calculate milligrams O2 liter into the amount of carbon fixed. You just need to multiply your value that you get by 0.698, and that'll convert it to milliliters of O2 per liter. And then you multiply it by another factor, 0.536, and that'll convert that to the amount of carbon fixed per liter. Once again, all of this is in both labs, the Biology with Vernier lab, and you can also find that in our inquiry lab. And those are part of the resources that you've been given. You have access to that lab. You can take a look at it. You can use it, whatever you want to do.
Melville, we've got a quick question.
Yes.
Is the initial sample in a lighted room and the same that's labeled as in light? Does that have a special light?
Oh, yes. Let me show you that. Well, you don't have to. You can use light, any type of light, but if you use a special type of light, everything is going to be better. So let me just show you this here. So this over here is the basic experiment that's been set up. I have our little water quality bottles in light and some that are wrapped in tinfoil in the dark. And the light that's being used is a new type of LED-based light that has blue and pink light, etc. And that's going to work a lot better than an incandescent, which doesn't have enough blue light in it. If you can put it in a sunny window, that'll work even better. So yes. Other questions? So if you use a good light source is going to be important, and I would not recommend using an incandescent light. With that, what I wanted to do is hand it off to Matt, who's going to talk about some really cool features, because You can also use our probes and his resources from Algae Research Supply. Let me go to that website really quick. You can see there's phytoplankton for everyone. He's got algae beads, algae culture kits, algae cultures themselves, and lots of other great resources. And we love working with him because you can actually use our probes with his algae cultures to measure photosynthesis and respiration in real time. So with that, what I will do is I'm going to stop sharing so that Matt can get ready to share.
Awesome, cool. Well, I'm gonna try to share my screen here. Share. I'm gonna do this one. So so hopefully you can see the screen there, and then also you can see my incredibly good looking face as well, so that you can see me dance around and point at things. So what I've done, if you guys can see, and John, I'm going to rely on you to give me feedback here if I'm on camera the right way. We have set up over here some algae beads, which are these fine things right here. These are algae beads that we manufacture. They're made from Chlorella vulgaris, which is basically food safe. And here's the Chlorella itself. This is going to be, this is a chlorophyte. It's in fresh water. It works really well for lab stuff. It likes to live in our laboratories. And when you're done with it, you can dump it right down the drain. There's no invasive species stuff at all because Chlorella is pretty much everywhere. And once it gets into the environment, it's basically food. So these are the strains that we're using. And we put them simply into a little beaker, a little plastic beaker. I like plastic ware in my laboratory because I hate cleaning up glass. As a parent, you know exactly what I'm talking about. And I have the dissolved oxygen probe and the pH probe inside here. Why? Because dissolved oxygen is a really great way to measure photosynthesis. Obviously, when photosynthesis occurs, one of the waste products is oxygen, and that goes into the water. Also, we can measure pH change as well. And these pH probes, as you can see, the data is going, is really nice. We're at the edge of where it's going to be effective. That way, you can see some of the little squigglies there. This is a really nice pH probe because you can get this granular with the pH. But the reason we're looking at pH is in water, carbon dioxide turns into carbonic acid. And in doing that, it makes the water acidic. So if pH goes down, then you have more CO2 present. So that's respiration. So what I've got behind me here is I started these algae beads out with some aluminum foil here in the dark. And we see both, there's a little bit of a negative slope here. That's oxygen being consumed. That's respiration. If I wanted to blow that up a little bit, this graphical analysis software is really great because you can expand that scale really easily and be able to do those things, to be able to see what's going on in a really granular fashion. And also pH is coming down again as well. Why? Because the algae that's in there is respiring and it's giving off CO2 as metabolic processes go on in the mitochondria. So at this point, when everything went up, and I'll turn this on right now too, when everything went on, we turned on a light. And so I've been playing, as John's been, and everybody else has been speaking, I've been turning on and off the lights today so that it's always cable management, right? So that we could see the bright light turned on. I changed the light there and I brought it to, first it was at 680 micromole photons. And I know that because the handy dandy, here we go. You can see the light meter there.
You're using our power sensor at the same time. That's great.
I am, because this is super important if we're going to be looking at photosynthesis. Why? Because algae, like most shade plants, is going to have there's a couple different levels we talk about with photosynthesis. We talk about on the very low side, if it's dark, you're going to see the respiration rate show up on your pH and your dissolved oxygen meters as a consumption. But then you're going to reach what's called compensation intensity, which is where photosynthesis is equal, the rates of oxygen production in photosynthesis is equal to the rates of respiration. That's when they're equal. In the ocean, that's at about 100 meters down in the water column, way, way out where it's clear in the ocean. And then everything above that is going to be positive photosynthesis, right? Where things are going in a positive fashion. And then we're going to reach what we call saturation. And that usually takes place around 100 micromole, between 100 to 150 micromole photons per meter squared second. And that's in what we call PAR, or photosynthetically active radiation, which is what this is measuring here. I'm going to slide this over and get a good measurement here. This is up at 800 or so ppfd, photon flux density, which is a sweet, awesome, sciencey thing. If you say that to your students, they'll think you're super smart. And that's just awesome. So I turn the light on, and then I put different levels of this shade screen that comes with the Vernier kits. All you have to do is get a bright light and then fold it over and over and over, and then just quantify it using their PAR sensor. And you're able to make a really cool P versus I curve, which is super awesome. So as John was talking earlier, I got it to 35, which is a little bit higher than the compensation intensity. But because this is an open container, the gases are going to have a chance to come to equilibrium. And that was right around here. Let me move that over. And finally, I put everything back into the dark. And then that's when everything fell down again. And then right over here, I was playing with it. And then I turned the lights on. As you can see, everything is just going straight up. This is everything that John demonstrated from his pond experiment yesterday, where he went out to Beaverton Creek, which, by the way, I've walked on before, John. And so he took the samples from Beaverton Creek and he put them into the light and dark bottles, wrapping them up. I believe John used the European version. He used aluminum foil. We didn't have the budget for that, so we just used aluminum foil here. But you can wrap these bottles up in this and do the same type of experiment. The reason why John's experiment wasn't as dramatic as ours is, I'll tell you why. It's because the density of algae that we have is a lot higher. So this is basically like saying, all right, you're going to put one person in a stadium and then look at the oxygen change, right? Or you're going to pack that stadium full of Taylor Swift fans and they're going to be screaming and respiring. And then those O2 levels are going to have to, they'll actually come down quite a bit because there's a lot of screaming that goes on for Taylor Swift fans. That was for Daphne. Anyhow, so that's why having a really dense culture is going to be a good thing to be able to make this dramatic and useful. And we're already off scale here, so I can correct that. Actually, Daphne, do you want to come and Daphne's a lot better at doing this type of stuff here. So now the one other thing we want to keep in mind as we do this is these are all SI units, right? We're talking about milligrams per liter of dissolved oxygen, and I'm sure pH is an SI unit as well. But one of the most important things when we're talking about photosynthesis is we want to normalize everything to biomass, right? So if you're going to be super, super nerdy about this stuff, you need to take the change in oxygen in milligrams per liter, and then make that divide that by the amount of biomass present. So you do that over grams of biomass or grams of algae. And since we want to do that in dry weight, because water is variable in cells, you'd want to dry the samples out. Now, we're not going to do that here, but that is something that we can do really easily with one of the little tools that we have, which is a Seshi stick, which we can, you can actually, we've got the Seshi stick, which you can calibrate to the biomass. So based on the Seshi stick depth, you can get right to dry weight really fast and really easily. And it just makes it super easy to be super, said super a lot, but be super quantitative, because there's one thing we like our kids to do, and that is graphs and math. And that way they can become super productive members of our engineering and science teams. But this is just a really simple thing. And as a matter of fact, Vernier makes a really neat spectrophotometer that would do the very same measurements as this would. And that's another fun thing that we've been playing with here in the lab as well. So this is basically what we'd call a P versus I curve. And Daphne will, there's one other thing we can do on this too. And if we can get this, can you grab the slope? This is really cool with this. So in grad school, I had to measure oxygen production, just like we are seeing here. Or if your students have taken those first year physics classes, you are going to be looking at acceleration or deceleration or anything in physics, which is a change over time. And this graphical software here can grab a section of that line and produce the slope. You got the, oh, it's right. Let me see here. And I think all we have to do is we grab this section and then we view statistics. Is it a curve fit or statistics?
You're going to do apply curve fit.
Apply curve fit. There we go.
And then it's going to default to linear and hit apply. Yep, there you go.
There we go. That's it. So this is really cool. This right here is like, I got to tell you, I had to do this on strip chart and paper. And this was back in like 2003-ish. So, but that was the best tech that we could come up with. And I could control everything. This is so much better. Now you have a slope, a literal slope here that you can see how many milligrams of oxygen per liter second are being produced. And then when you normalize that to the amount of biomass, all of a sudden you're at like, oh, I can make predictions about the ocean now, knowing how much biomass is there in the ocean. And your students just become super, super, super dangerous with their predictive abilities. So I'm super excited to have these in the lab and be able to do this kind of analysis with the little slopey things there. So yeah, that's how we've been using it in our classes. We're teaching our APSI classes this year using these Vernier instruments. And you can do this, we have this, now that we've grabbed a bunch of data. These are our algae beads we've done with it. When it starts out, it looks like this. After they've been beat up with a stir bar, they look like this. Some of the cells come off. But you can see there's a lot of biomass in there that we're playing with. And that makes the difference into making these really dramatic and being able to use them in the classroom. Yeah, and that's why I like this stuff. It's pretty cool. Any questions? There might have been a couple that had come out there.
I don't see any, but one of the things that I thought is also really amazing about Algae Research Supply is they don't just have chlorella, they have different types of algae that you can take a look at. Spirulina, which I didn't realize likes alkaline water more than it does neutral, and even look at other algae as well that are in, saline environments versus freshwater. There's a ton of stuff that you could do just right there also in your ecology class or when you're teaching ecology by looking at what type of nutrients do algae need to grow, how are they going to grow, etc., and how are they different. Yeah.
Oh, you know what we could do too? Let me, this is another thing. We've been working with brine shrimp because, between me and John and Sarah, we think algae are pretty awesome, but algae is hard to get other kids to kind of drink the Kool-Aid there to make it look really neat. So we brought in brine shrimp because brine shrimp eat the algae, and then we can quantify the biomass of the brine shrimp and look at the grazing rates of them using our Seshi stick again. But a neat thing you can do with all this instrumentation is you can look at how fast the brine shrimp consume oxygen. You can put 1, 2, 4, 8, 16 brine shrimp into water and observe them consuming oxygen as well. And that way, we can kind of make a balance of how much plant material is necessary to support the animals. So it's just another fun thing to do. But by bringing those animals, those grazers into the equation, like we have in our beaker bags and stuff here, having these animals here eating the algae, we make these kind of ecosystems, and the kids really get engaged. They really see that that they can actually come in, check on their algae and stuff, do the quantitative measurements every day, and it keeps them engaged in learning and just being curious about everything. Because brine shrimp are really easy to keep, and those beaker bags, they keep for for years at a time in the classroom. All you got to do is keep them wet. The light measurements, I saw your note there, Fiona. The light measurement is their Go Direct PAR sensor. I've got it right here. It's really great. I believe it's waterproof, isn't it, as well, Jonathan? Yeah.
Yeah, it's waterproof. Like you can actually put it down in a water column. I can show
Yeah.
people all three sensors. If once everybody's good, I can go to our website and show them like these are the three sensors that we're using. Oh yeah, it would be okay in 35 ppt. It's meant to be put in salt water.
Awesome. We haven't submerged ours yet, but that's something that we would do in the algae production world. We would use light sensors. An algae production pond is only like 20 centimeters deep, but there's so much biomass in it, so that the euphotic zone, the light that's above the compensation intensity, is only going to be the top like 3 inches or about 6 centimeters, right? So you've got only this much that's in the euphotic zone, and mixing keeps the algae going into the light and the dark. And how I would, when we're quantifying all this stuff, is we would take light meters exactly like this, and we would put them underwater and create a depth profile of the light over time. So this is the same technique that you would use in your pond in limnology or in oceanography in the classroom to look at what the light intensity is going down. So with this sensor, you'd want to make sure I believe it's called a cosine sensor. So it's only measuring light in this direction, and at a tangent or whatever it is coming in from the sides. But in oceanography, we're looking at light that would go down and hit a particle and come back up. So we'd use a spherical 4 pi R sensor. But this one is absolutely dynamite for doing just basic submersible light measurements.
Yeah, it's cosine corrected, and then I think there's a lab, I believe, on the website for doing just that, where we tell you can attach it to a Secchi disc. You can attach it to a Secchi disc and then just lower it into the water column up to, I believe, around not quite to 5 meters, but around 4 meters.
Okay.
To see how the light level changes.
Good to know, because I think that's probably the length of the cable too.
Matt, we have a great question from Kim.
Sure.
Kim says, I'm using a beaker bag, but the instructions say to add algae flask, but I don't see where to add the beads or culture.
Okay, so there's this might have been for an APSI. The algae beads are a separate project. They're to demonstrate. We have them in this is what they're talking about. We have these algae beads in little vials with a pH indicator solution, so that you can see respiration if it goes yellow and photosynthesis if it turns red. And you could do these as a separate project, putting them into a bright light or putting them into dim, hot or cold. Anything that's going to affect metabolic rates of algae, you can see with that. But with the beaker bags, again, that's a whole different topic that we haven't talked about here. But basically, you add the algae culture salts. You basically want to take care of the physical, chemical, and biological components to grow the algae. The physical being water and salts, chemical being nutrients, and biological being the algae culture. And then you put all that together in the beaker bag in the presence of photons of light like we have up here, and the algae will grow. Usually you're going to have about a 1 to 3 day lag phase where the algae is not going to do too much, and then it's going to grow exponentially. And that's where it gets really fun, because the algae that we use, the Nanochloropsis, will grow so fast that you can measure it every 2 hours with the Secchi stick and be able to get a slope.
We have another great question, Matt, and that is, since you're showing us a live reading, the question is, compared to the lab that we have where the kit, the bottles get sealed up and left overnight, you're doing this actually in an open container, correct? Can you tell us a little bit about that and how fast it is?
Yeah, so these open containers here, they're going to be super duper fast, especially if you use the algae beads in there, because the algae beads have concentrated chlorella vulgaris. We actually spin it down and concentrate it in there. And so that's going to be a really fast response. And the reason that it's fast is, again, what John and I were talking about, which is that there's a lot more biomass there. Just the sheer amount of biomass that's going to be even in these 2 containers here, you can see the difference. You can see through this algae, this is chlorella, versus this. You can't even see through the water there because there's so much biomass in these algae beads. That's why this is nice to see, this very dramatic curve here. dramatic and, you know, nerd talk. It didn't like go and wave its arms around and complain. That would be my daughter.
Yeah, one of the things to remember here is because Carolyn asked, like diffusion out is not a problem. It's not going to be a problem because it's happening so fast. That's one of the things I tell people a lot is if you want to show photosynthesis and respiration, it's great. And look at oxygen. It's great to use dissolved oxygen because the amount of oxygen that's actually in water is not nearly as much as you would think that it's like actually in the atmosphere, right? Where it's 21%. So it's much easier to see changes. So you can actually use an open beaker and look at, like, you can throw some yeast in there too, and it will dramatically take up all the oxygen in there, or use algae and show that you can put it in light and the oxygen level will go up, but in the dark oxygen level will go down.
You know, can I speak to that too, John?
Yeah, of course.
This is absolutely the best question because I struggled with trying to get this demo set up so that it would be something we could use. When if I was doing this for publication, I would absolutely have that entire chamber closed with no air bubbles in it whatsoever. And prior to doing any work in it, I would bubble nitrogen in there to drive off any oxygen gas. Okay. That's a really good thing to do. However, in most teachers' lab, you don't have O2 gas available to you. So what we want to make sure we do is we want to make sure that we're going to keep the dissolved oxygen level below saturation. Saturation for most of us, depending on what altitude we're at, is around 8.5 milligrams per liter. Right now, we're above 8.5 milligrams per liter, and we're going to see stuff like this going on. If you can see my beaker bag here, those oxygen bubbles right there are exactly that. This solution is super saturated and the oxygen's coming off and getting driven off. And as a matter of fact, if we were to leave this water on here, we can see up now on the top, it's at 11 milligrams per liter. That is going to asymptote. And that's because we have the stir bar in there. And even though the rate of production of oxygen is still very high, it's being driven off. So what we want to do is we want to, you can exhale into it to drive the oxygen down. Do you want to do that? No, Daphne has refused the oxygen driving down concept. But if you wanted to exhale air into it, you can drive your O2 back down to below. And I like to start it off. You could either put it in the dark, and that'll consume the oxygen for just like an hour or so before the lab. But you want to keep it below that 8.6 or so. And that way you're not going to see these crazy things because you'll see it asymptote here in a few minutes.
All right.
Good question.
We've got two more great questions. The first one is an algae culturing question. Can we grow the algae in a new water bottle containing reverse osmosis water?
Yes. Water bottles are great to grow algae in. We use them all the time. If you're going to do that, RO water is okay. I like either well water, because well water has lots of minerals in it, or spring water if you get it bottled. Avoid tap water because it's either going to have chlorine or chloramines in them. Chlorine you can drive off by leaving it out for a couple days, but chloramines are evil and they stay in the water, and they don't even come out very well with carbon filter. But you can absolutely use other sources of that. But what you'll need to also grow algae, you need to make sure that you add some salts to it. And these salts we have them in little baggies pre-measured, and those are trace metals and other stuff. We basically put baking soda so that there's a carbon buffer, a little bit of sodium chloride, maybe a little bit of carbon, magnesium. But it's the nutrients that are going to be the most important thing. And we have these already pre-measured and pre-weighed. These are from a different thing here, so that you can add one little baggie of salts and nutrients, and you can dump them right into a 500 ml bottle of water, and that's algae culture media, and you're ready to grow however you want to do that. And we have saltwater media, freshwater media, and as John had mentioned earlier, the spirulina is an alkali media.
Excellent. All right, our second second question is about the PAR sensor. I answered half of it, which is the regular PAR probe, the original BTA style, the LabQuest style. Can that go in the water? The other part of that, though, are your PAR readings, are you taking them in the water right now?
negative. That's a good idea. Ooh, but let's do that, shall we? That'd be really exciting. Let's watch this. Can we start a new one, and then I'm gonna do this would be a depth profile, but it's kind of different because I have the light on the bottom of the algae here. We'll see if the probe is waterproof right now, John. Here we go.
It's waterproof.
We will know. Do you ready? Daphne's getting the data logging ready. I'm going to take the light bar out there. It's going to just create noise in the signal. Okay. So are we on, Daph? Okay. So here we go. There's it's rather stiff there. Okay, this is at the surface of the water. And as I go down, you might want to do
as you go down, it's going up. You're at like 100. You were at 100 when it's pointed directly at it, and then you brought it back up. So it went from
redo the time scale
12 all the way to 100, and then back down to 12.
Yeah, we're at. Let's get all the way down here because at the very bottom of this light that we have here should get really high.
Oh, there you go. Yeah, it
500.
It's around 500. Yep.
Yep. So the neat thing about that is that down at the very bottom, the algae is going to be deploying some photoprotective mechanisms because that's too much light. It's above their saturation intensity. So they're going to be putting some pigments out there that will protect them from, oh, that's really cool. Yeah, let's do it again. Sorry. So we're looking at the pink line, and this is the surface, right? And that's down. This is about we're probably double the saturation of the compensation intensity at around 30 micromole photons per meter squared second. And then as we go down in the water, it's going up, 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 up. And right around there, that's probably saturation around 100 micromoles. But as we get down to right next to the light, we're getting up into the, oh, wow, I got to like 900. That might have been right next to an LED. But there you can see that that's the water with the algae in it attenuates or reduces the flux of photons into the water. And that light is absolutely absorbed by the cells. And that's what photosynthesis comes from. So this is why in an algae culture, you want to keep things mixed so that the cells that are in the dark can get to the light and then go back to the dark. Because they'll be like, oh my gosh, feast on the light. And then they have so much of it that they need to rest. And then they go to the dark. And then if the culture is really good, it'll be going light, dark, like that. I get nerdy about that stuff. That's like a really fun physics question about how you mix the algae culture.
I know there's so much that you can do with this. I remember I was playing around with this and researching a little bit, and I finally, like, in my mind, finally clicked for like why losing the ice shelf up in the Arctic is going to be so bad for polar bears, and that's because a lot of the primary productivity that's actually happening is happening over the continental shelf, and as that ice recedes and recedes and recedes, it's getting more over the open ocean, which means the polar bears are like swimming all the way out to the ice that's over the open ocean. Not as much primary productivity, which means there's also not going to be as much productivity of other things, fish, seals, etc. So they have to travel a lot farther just to find food. So it's a really great way to explain to your students, like, this is why this is important. Or if you're like me, I used to study Cnidarian algal symbiosis, or I was really into it. Would talk about it a lot. That's another reason why with this light level, that's why reefs only really exist in relatively shallow waters and aren't found at super, super deep places. Some are, but the really giant coral reef systems that we're all talking about, or that people are talking about, typically happen in relatively shallow water where there's plenty of light and they have a algal symbionts with them. When they bleach, that means they've lost their algae. So there's all sorts of really cool stuff that you can do here. All right. Any other questions?
Nothing more waiting in the chat at the moment.
Well, it's so great that you guys, everybody that's watching the videos and the folks up in Beaverton, you guys are amazing for taking the time in your summer vacation to come and learn a new thing, to wield algae as a tool and to learn how to better use this equipment in your classroom. It really shows your commitment to teaching the next generation of scientists and engineers. Because goodness, we need smart people right now. We've got a lot of problems that we nerds are going to have to clean up. So thank you.
Yeah, there is a
Matt,
great question that was. Go ahead, Sarah. You're going to ask.
Oh yeah, there's one more question, Matt, and it's one that I know you're going to love answering. It is there a consumer that you can add that would be something that you could measure density with a hemocytometer?
A consumer? How do you mean by with a hemocytometer?
I think,
I'm thinking your brine shrimp.
oh, he says besides brine shrimp.
Oh, besides a microbe.
Microbe.
Yeah, is there any type of maybe zooplankton that
that would eat
could
the
be added
algae as another grazer,
or
oh my
something
gosh,
like a copepod?
Yeah, copepods are great, and we have them here in the lab as well. We've got a couple different strains of them. The thing with classrooms is copepods don't male well, and they don't keep really well unless you give them a lot of love. Brine shrimp, on the other hand, they cyst up, so you can ship them in a plastic baggie, right? And the cyst, and then we ship those with our Brainy Briny kits, and they're amazing because you get the algae culture growing, you add the brine shrimp cyst to it, you get larvae in like 2 days, everything works great. And we've got like probably a 99% success rate with getting the brine shrimp to hatch and grow. So it's really easy. It's the best system that we could come up with to bring a primary producer and a grazer into the laboratory or into the classroom. And
you could easily measure the amount of productivity with our DO probes by using those brine shrimp as well, even though you wouldn't be able to use a cytometer, but that would be super easy. And that actually is great because that was another lab that we've talked about doing together. So if you think that would be a great idea, let us know at biology@vernier.com or just let us know in the chat so that we can tell Dylan that we want to do that next year. That would be great.
Yeah, let us, if you guys have any other ideas too about different ways that algae can be used with the Vernier probes, please let these guys or us know. Vernier's been nice enough to loan us some of the equipment here, so we can play with it and do little videos as well. So, and it's super fun because this stuff is really cool. And the displays on it work really well, so it makes it really easy for the teachers to use.
Yeah, and if any of you are going to be at Able, I'm going to ask Matt to send me some algae to Able because we're going to play around with some algae cultures at Able. But if you're at Able, come and see us. Able is the American Biology Laboratory educators meeting, and that's happened in Maryland. We saw Matt there last year at UC San Diego, and hopefully we'll do a workshop together next year, wherever it is. So thanks so much for coming to this webinar. I'm glad you all thought it was great.
Take care, guys.
Thanks, everybody.
Cheers.